Hello students, already two experiments over. So now I am going to demonstrate that effect of pH on salivary amylase. You know that salivary amylase is active at optimum pH, that is 7 or 6.8, but it remain inactive in low pH or high acidic condition and high alkaline conditions. For this experiment, you have to take materials like test tube stands, 3 test tube stands, one for pH 4, one for pH 7 and third one for pH 8. Then test tubes, pH solution, ready-made pH solution also available. So, take pH 4, pH 7 and pH 8 solution, then iodine solution, 1% starch solution and 1% sodium chloride solution. Sodium chloride and starch solution how to prepare already explained. Then dilute salivary amylase. How to prepare? 1:19. Okay. Then measuring cylinder, funnel and cotton wool, that is for saliva collection. Theory. Salivary amylase present in our mouth is active at temperature 37 degrees centigrade and optimum pH 6.8 or 7. At low pH and high pH, it is inactivated form. So, generally when you carry out experiments, so that starch will not get digested in pH 8 and pH 4, but get digested in presence of pH 7. So, in experiment we will carry out, this is the observation table, that is serial number, time in minutes, pH 4, pH 7 and pH 8. Under this pH, what are the color changes occur, that you have to observe. So, let to start. Now, I will start experiment. You have to arrange 3 test tube stands with 6 test tube and each with 1 ml iodine solution. First make ready, one for pH 4, one for pH 7 and another pH 8. Then there are starch solution, 1% and 1% sodium chloride solution, and there are 3 test tubes that beaker, one contain pH 4 buffer, another pH 7 buffer and next one pH 8, and this is salivary amylase. So, you have to take 3 test tube and label each one with pH number. This is pH 4, pH 4, pH 8 and pH 7. Make it as pH 7. Then take 5 milliliter starch, pipette out 5 milliliter starch and add in each test tube 5 milliliter starch. This is pH 4, add 5 milliliter starch, and 5 milliliter in pH 7 test tube. Okay. Next, you have to add 1 milliliter sodium chloride. So, sodium chloride generally activate our amylase. this work is over. Next you have to take water bath. The temperature you have to maintain 37 degrees centigrade. Next you have to add respective pH. This is acidic, that's pH 4. Take dropper or measuring cylinder or graduated tube. You have to add 1 milliliter in test tube 4. Remember one thing, you don't change the test tube, that dropper, because slightly mixing of this dropper with the other pH will not give result. This work is over. Then pH 8. Up to 1 milliliter you have to add. And keep this dropper in this test tube. Then third dropper take, and you have to take pH 7, 6.8 or pH 7. You just add up to 1 milliliter. Okay. Now salivary amylase 1 milliliter in each test tube. So first we will carry out 8 pH containing 8 pH 8. So one by one experiment will carry out and take it as set for pH 8. Add 2-3 drops, or it will turn to blue black color. Next pH 4. The first test tube, then pH 7, and third set, and wait for 2 minutes. After 2 minutes interval, again we will continue the whole process. So after 2 minutes interval, add solution from pH 8 to the second test tube of the set. Then pH 4. And pH 7, the second test tube, and wait for another 2 minutes. Look here, it is almost same color, pH 8. This is pH 4. I'll turn to blue black color, and this is pH 7, also blue black color. Again, after 2 minutes interval, we will continue the process. And from 7 to the third test tube. So look here, all three that pH 4 remain blue black color. Here also pH 8, same color, deep brown color, and pH 7 gradually, look here, gradually color is changing. First blue black, deep brown, then light brown. We will continue the process. So this is the process. You have to, with 2 minutes interval, you have to carry out the experiment. pH 7. So it is almost iodine color. So when you continue this process, here that pH 8 all will turn to blue black color, and pH 4 also all will turn to blue black color. Look here, all are blue black color, but gradually there is a change in pH 7. So look here, this is 0 minute, 2 minutes, this is 4 minutes, 6 minutes, and 8 minutes. It will remain as iodine color. That means it takes 8 minutes to digest starch. So here pH, optimum pH required for digestion of starch, that is pH 7 or 6.8, where amylase becomes more active and digest starch. But in low pH and high pH, it remain inactive. So this is the experiment for effect of pH on starch. Thank you.